This is called a translational grant. So she's been given funding for 3 years to get to her milestone, which is the cells in GMP facility. Okay, so our phase 2, yes.
Where did they get their funding from?
From CIRM, California Institute for Regenerative Medicine.
Okay.
Yeah. It's always a tricky process. We can discuss funding. It's challenging. So for the phase 2, preclinical and clinical studies, we want to do a small clinical trial of iPS cell-derived dopamine neuron transplants. And it's obvious, although I don't state it here, that we'd like to transplant the cells to the very people that we get the cells from, because we know they'll be a perfect match. In the phase 1, of course, we get the cells to GMP. In phase 2, we're going to need additional funding, either from the NIH, who may be willing at that point to fund another clinical trial, commercial funding. We'll need new expertise because a clinical trial is something that different people than us do, and we'll need clinical partnerships. The FDA requires us to do preclinical safety studies, a new drug application, and a safety trial followed by a dosage / efficacy trial, and then finally a larger trial with phase 3.
So the safety trial will be what we're going to embark on? Is that called the safety trial, or what would you define as a safety trial?
Well, so the trouble is you have to do, so the first thing you do is you transplant the cells into animals and make sure it doesn't cause them any harm. So that's the first preclinical safety. That's preclinical. And then the clinical safety is actually putting the cells into people, but you do it at such a pace that you can actually see whether it has any ill effect before you put them in another patient. But that's actually a patient study. So you put them in a small amount,
like you take one of these 6 chosen patients where you develop the cell line, and you just take one of those patients and put a very minute amount.
No, not necessarily a minute quantity. We could put the amount of cells that we think will be efficacious.
Okay.
For the safety trial, we want it to look as much like a real treatment as possible. The difference is that we have to, there are lots of things we have to do to make sure that those cells don't harm the person. Yes?
All of this works, the funding comes in, and everything is successful. How long does it take for that to be available?
Okay, so we can control the first part, the phase 1. That's 1 to 1.5, maybe 2 years at the outside. This is going to be dependent on where the field is at the time we finish our part, and whether there are clinical trials being done by a company, for example, that we can get involved in. If we have to start from scratch, because Jeanne already has gotten approval for a clinical trial for a spinal cord transplant, we could follow in their footsteps. It's expensive, but we know what to do. And then it would take us another 3 or 4 years.
So, like 6 years altogether?
Outside, yeah. Yeah.
Yeah. No problem.
Yeah.
How expensive is that? You said it's expensive.
This part?
No, after the phase 1.
Oh, per patient? That will depend on a lot of things, as you can imagine. Whether insurance will cover it.
No, I'm sorry. Let me ask you said that getting to the phase that, like they're doing.
Oh, I see. Right. The phase 1, we've estimated a minimum of $300,000. I mean, that's our real cost.
Yeah.
And we're donating the labor, so that's how we want to get through the first part. The second part will cost at least $1 million, if not more.
Okay. Yeah.
And that's where it's sort of like you throw the money into the,
right,
into the trash.
And then that takes you to the 6 years
That's
that you're
right.
talking about.
That's right.
Now, can you apply for FDA approval at the time you start phase 1, or do you have to? I mean, can that process begin at the very beginning of phase 1?
They have to approve this in concept. We have to show safety in animals before we can start a phase 1 clinical trial. So it involves more paperwork than it does experimentation. But it's for the good. I mean, every single drug and every treatment has to go through the same procedure. That's why I say other people need to be involved. They're called regulatory people. Okay. We're called scientists.
Okay.
Okay, so now I want to get to the part that you haven't heard, none of you have heard before, which is going to be a series of very short talks by people from my lab. This slide is an overview of all the things that we do in this lab group. And it includes basic research and translational research. A lot of this work is in basic research supports the applications of translational research. So the subjects that we had requested are a short talk on our Alzheimer's disease project, our autism project in which we use Fragile X mutation, which causes autism, our ALS project, our multiple sclerosis project, and our reprogramming of endangered species. It's a collaboration we have with the San Diego Zoo. So these are the people that you will be hearing from, and they're sitting over there. First, Kit Nazer, who's my graduate student, will talk about neural stem cell transplants for Alzheimer's disease. In this case, the stem cells are not intended to replace the cells that died, but rather feed the cells so they make more synapses. And this is one of those projects that we like to say we've already cured a lot of mice. So this, the Fragile X autism, Kit will also talk about this. This is an earlier stage project in which we're going to try to find out what causes autism, but by doing cell culture. INBAR's Endangered Species Project is using iPS cell technology to help rescue endangered species. The ALS project, Ying will talk about using molecular, about using molecular biology techniques to correct mutations in cells, which is a method that is not only applicable to ALS, but lots of other diseases. And then finally, Ron, the graduate student, is going to talk about using cells that come from iPS cells, neural progenitor cells, for treatment of multiple sclerosis. Okay?